Okay, so this is a lab video for the Biology 1 cell lab. I'm Dr. Ruiz. And I've got some information on here I want to talk about first, and we'll go through some of the slides on the PowerPoint. This is a Practical 1 lab PowerPoint. The cell lab, we would normally for this lab be looking in the microscope at images like bacteria or diatoms, amoeba, paramecium, and hopefully it would help you learn the names and kind of get a rough idea of what they look like. And then if we did this lab practical in the lab, you would look in the microscope and have to identify these in the microscope. So when you take the practical, instead of looking in a microscope, you'll be looking at maybe a figure from this PowerPoint and telling me if it's the diatom, paramecium, tardigrade, whatever organism it might be. So the first thing I thought, let's group the two main categories of life, right? Now, we've talked about this in class, I think, already. Prokaryote versus eukaryote. Okay, so a prokaryote are cells or organisms that do not have organelles or nuclei, right? They go in this category. There's a domain called bacteria, and there's also another name, another domain, but there's one domain called bacteria that the bacteria fall into, as well as Anabaena and Oscillatoria, which are types of bacteria. Okay, they're not your classic bacteria, but they are types. So they belong in that domain. Eukaryotes have organelles, and they have nuclei. They belong in the domain Eukarya, right? There's actually 4 kingdoms. So I might give you the information about which kingdom. You know, a plant belongs to the Plantae kingdom, right? Animals belong to the Animalia kingdom. All right, but all these organisms, diatoms, amoeba, paramecium, tardigrade, are in the, they're eukaryotic organisms. They belong to the eukaryote domain. So, bacteria, these are images of coccus, bacillus, and spirilla. Some bacteria have these, either they're round or they're rod-shaped or spiral, right? And the bacteria belong to the domain bacteria, so they have the same name and common name. Then there are a couple other organisms you should be familiar with. Anabaena, right, or belong to the domain bacteria, the heterocyst. These are actually long chains that each one of these little rectangles or squares is an organism. They just kind of stick together. And that's Anabaena. And we have, right, Oscillatoria. They also belong to the domain bacteria, and this is an image of them. We've got slides of these. You'd be able to watch them. I'll look at them in a microscope if we did this in the lab, but obviously we're not doing that. So then, a diatom is an organism that belongs to the eukarya domain, the kingdom Protista. Right? Protista are single-cell organisms. Even Volvox is considered to be in the kingdom Protista. They have these big, long, these are like structures, and then there are individual cells inside them. They're pretty, Protista is a pretty complicated kingdom. They're even thinking about turning them into several kingdoms, and they might eventually do that. So, right, when you look at this, this is pretty complicated too. Each one of these guys are one organism, one type of diatom. These are another type of diatom. This is another type. So there might be, you know, 4 or 5 different diatoms actually in that one image. Then an amoeba is a eukarya. In the eukarya domain, it's considered a eukaryotic organism. And in this figure, you can actually see the nucleus, right? That's the nucleus, and then this is the cytoplasm. So the shape of this organism can vary. It can be all sorts of different shapes. It's not consistent. When you take this for the practical, I'll tell you to recognize this type of organism by the color. So sometimes the colors can help. And that's just the color that they turn that organism into. When they put them on the slide, they give them a color. Like this one really doesn't have a color per se. It's not been stained. Right? This would be the water that it's in. This is an organism. This is another one. Here's one down here. And this is called Paramecium. You probably heard of it before. It's a single cell organism like the amoeba are. So they're about, bacteria are a little bit smaller than these guys, but these are pretty small cells, right? If you don't have a microscope, you can't see them. And then they have these hair-like structures that you can see pretty good on this image. They're called cilia, and when they move them, it allows them to kind of swim around using their little cilia. These cilia, they can bat. They kind of wave. You can see them. They're like wave-like motions. And so that's Paramecium. It's a eukaryote, kingdom Protista. Now tardigrade, one of my favorite animals. Another name for it was water bear. Okay, that's a common name. Tardigrade is the genus. So these are water bears. They have, looks like 8 legs. And it almost looks like they have claws too. But they're really tiny. They're bigger than they might be about the size of a Paramecium, maybe a little bit larger. Paramecium are pretty large. But these aren't single cell animals. These are, multicellular. I don't know exactly how many cells, but there isn't too many. There's probably 1,000 or so cells in a water bear, maybe less. And these are the guys that made it to space and came back, and they survived. We always think that if if they didn't wear little space suits, that they wouldn't survive. But somehow these guys can freeze. They can live in pretty warm temperatures, much more than other animals. So they're very hardy. But they belong into the Animalia kingdom. Now yeast, they're eukaryotic too, but they're much smaller. They're single cell organisms. Each one of these little red is a yeast cell. Here's some more pictures. There's probably a better picture of it than this. These are on a slide, and they stained them red. This is an electron microscope picture of them, which is much more detailed. You can kind of make them out much better. They look like they're on top of one another. They belong to the fungi kingdom, and Saccharomyces cerevisiae is a genus and species name given to one particular type of yeast, right? This is, I think, the yeast that you use for baking yeast. You buy this, you could buy that in a grocery store. When you put it in bread, dough, right, the purpose is the yeast in the dough will make carbon dioxide, and that's why the bread expands, or rises is another. So these are white blood cells, or leukocytes is white cell. Erythrocytes are red cells. So the red ones are red blood cells, RBCs, and then these are nucleated white blood cells. So if you did a blood smear, this is what it would look like, right? But these are human blood cells, so we're in the kingdom Animalia in the eukarya domain. Now, this is a plant cell. So normally we differentiate animal cell versus plant cell, right? And plant cells tend to have a chloroplast and appear green in color. Not always, but most of the time. Right? The green is from the chlorophyll in the chloroplast. That's the chloroplast is the site of photosynthesis, and they're just basically little discs. And because there are cells on top of cells, it's depending on where you are focused. You know, these chloroplasts might not be in this cell; they might be on the cell on top. So it's kind of hard to, you know, to interpret what you see there. This is an onion, which is a plant cell, kingdom plantae, just like the Elodea, right? Elodea is the name of the plant. That's onion. Cheek cell is a good example in lab. We would take a cheek swab and get your cheek cells, paint and color them blue like this, right? They belong to eukaryote domain, animal kingdom. You can see the nuclei. There's one nucleus, and then the cytoplasm of this cell. You can see that in this image. You know, that image is blue to help you see that. So organelles are found in eukaryotic cells. They're not found in bacteria, you know, or prokaryotes. So we'll talk about the mitochondria, endoplasmic reticulum, nucleus, Golgi body in the lab. And I'm going to show you a plant model in a minute, and we'll go over some of these in the model. So in the plant, right, we have, this is, say, the cell wall around the plant. Inside the cell wall of the plant is the cell membrane. Think of that as maybe the yellow line that goes around. And then this big, large is not the nucleus. This guy is here. This is called an organelle. The central vacuole is filled with water. Right, so this bracket is for the central vacuole. This is the Golgi. Not the Golgi, this is the endoplasmic reticulum rather, in blue. This is a mitochondria, the orange guys. And the green disc, this is a green disc cut open. That's a chloroplast, right? All these green are chloroplasts. And then this whole oval is a nucleus, and the center thing here is called the nucleolus, which is a structure inside the nucleus. So that's a plant cell. The animal cell model, right, contains, you know, this is a circle. This, the teal color here is the plasma membrane of this cell, right? This is the white. This big circle here is the nucleus. And then we've got the endoplasmic reticulum in blue. We have the mitochondria are orange like they were in the plant cell. There's no chloroplast in this. Right, this whole bracket here is for the white nucleus. It's kind of cut open. This circle in the middle is the nucleolus. You have that in the plant and animal. This is the nuclear pore, this little circle, which is a hole that allows things to get into the nucleus and come out of the nucleus, go in and out from the cytoplasm to the nucleus and back and forth. And then this black, this is actually a structure, not an organelle. All right, that's the centriole. Okay, and that's the parts of the animal cell model, which when you take the practical, I might expect you to know the different parts or the different organelles found in the animal and plant cell. So another thing to mention about this is that when you describe a solution that has cells in it, so these are supposed to be red blood cells, the solution and the solution that's in the cell itself are two different solutions, right? So if the cell concentration in both is equal, we say that it's an isotonic solution. For a red blood cell or most animal cells, that'd be normal saline, is another way of thinking of it. If it's a hypotonic solution you put the cell in, then the solution has hypo, which is low salt concentration compared to what's in the cell. That causes more water to go in the cell and it lyses. If you put a red blood cell or an animal cell in a hypertonic solution, hyper meaning hyper salt, then it shrinks the cell because the water, more water gets out than goes in over time. So you call that a hypertonic. Now, it's a little bit different for plant cells. And then this is a general statement. Not all plant cells are like this, but for the majority of plants, they want to be in a hypotonic state, right? The solution they're in has lower salt than what's in the plant cell. And that exerts pressure. You have a cell membrane, and on the outside of the cell membrane is cell wall. So that's kind of, when we say there's pressure, we're kind of pushing against the cell wall. Whereas in an isotonic solution, you lose that pressure, right? They describe it as being flaccid, or you might say the plant might look wilted. And it has something to do, it can have something to do with the salt concentration, or just not having enough water in the central vacuole could do that too. But if you put a plant cell in a hypertonic solution, which we actually do in lab, we were doing the lab under a microscope, these chloroplasts that you see here would normally be all spread out in the cell. What happens is when the cell membrane shrinks down and the water from the cell gets into the space between the cell and the cell wall, then you concentrate all the chloroplasts into the center, or not necessarily the center, but they all come together because the membrane is closing in on them. And if we did this in lab, we'd add the hypersalt, the hypertonic solution, and we'd slowly watch this occur. It takes 2 or 3 minutes to say go from this kind of look where they're kind of all spread out to where they are concentrated. That's referred to as plasmolysis in the plant cell when that occurs. So it's a little bit different than when an animal cell lyses, it occurs in a hypotonic solution. And when you put a plant cell in a hypotonic solution, it's normal for them, but you get plasmolysis in a hypertonic. The cell doesn't actually lyse like a red blood cell. A lyse in a red blood cell explodes. That doesn't occur in the plant cell. So we kind of went through turgor pressure, hemolysis, crenation, which is crenating, meaning that the animal cell shrinks. Isotonic, hypertonic, hypertonic. So these are words, vocabulary you should be familiar with for a lab quiz and for the practical one. Okay. All right, so that's it for this video. Study hard.